Hi everyone, my name is Meredith Rogers and this is an overview of my research project investigating the relationship between Fusobacterium nucleatum and neurodegenerative disease. Before I begin, I just wanted to thank my faculty mentor, Dr. Yiping Han, and all members of her lab for their support throughout the duration of my project. Fusobacterium nucleatum is an opportunistic gram-negative anaerobic bacteria that is commensal to the human oral cavity and increases in prevalence with disease states. The pathogenicity of FN is thought to be derived from a novel adhesin protein FAD-A, which is responsible for the bacterial attachment and invasion of host cells and extraoral sites. The extraoral colonization of FN can result in systemic human diseases such as adverse pregnancy outcomes, colorectal cancer, and rheumatoid arthritis. In addition, FAD-A is thought to be associated with the bacteria's ability to cross the blood-brain barrier and polymerize into an amyloid-like protein. These amyloid-like proteins are a hallmark of neurodegenerative diseases, suggesting FN colonization may contribute to the development of such diseases. Therefore, the purpose of the study was to elucidate the potential link between the extraoral presence of FN and neurodegenerative symptoms. To begin, 3 different strains of FN were cultured in our lab: FN 12230, which was the wild-type strain, US1 FAD-A deletion mutant, and LAM, which was the secretion mutant. Each strain was plated and incubated in an anaerobic chamber at 37 degrees for 48 hours. Single colonies were picked from each plate and cultured in Columbia broth for an additional 48 hours. The resulting colonies were then resuspended in PBS once they reached the stationary phase for cranial injections. This study was conducted on a total of 46 mice, which were 8 to 12 weeks old at the start of the study. 22 of the mice were cranially injected with 3 different strains of FN. 14 other mice received saline injections, which served as a control for the cranial injection, and 10 mice received no injections and served as a control for the study. After a 3-week surgical recovery, behavioral testing began, which aimed to test the memory and sensory motor functions of the injected mice. The first behavioral test, known as the rotarod, consists of an accelerating rod that increases from 4 to 40 RPM over a 5-minute period. The time spent by the mouse on the rod before falling was recorded for each trial for 10 consecutive weeks. The second test, the Barnes maze, consists of a circular table with 19 holes chosen to be false holes and 1 true escape hole. Each mouse was trained for 1 day to find its escape hole and then tested for 4 trial days each week to assess the mouse's latency time to reach the escape hole for a total of 3 weeks. The results of the rotarod experiment yielded inconclusive. It was thought that the variability in this data could be explained by the fact that sensory motor abilities of mice may require more than 10 weeks to improve. However, there are several trends in the data that can be further investigated with a larger sample size and more data collection times. For the second behavioral experiment, the Barnes maze showed an overall downward trend to locate the escape hole across all treatment groups during the first week. However, male and female FN-injected mice showed a similar trend in which their latency times to the escape hole remained relatively high across subsequent testing weeks. This suggests that long-term memory of the mice may have been impaired. In conclusion, our data suggests that FN has the potential to elucidate neurodegenerative symptoms in mice by impairing long-term memory. Future directions of this study include using brain imaging to verify the presence of bacteria in the mice and utilizing ApoE mice to determine whether FN may worsen existing neurodegenerative symptoms or produce the conditions de novo. Thank you for listening.